Welcome. In this session, we'll discuss the physiology of neurosensory retina. In the previous session, we have discussed the anatomy of neurosensory retina, and in the next session, we'll discuss the anatomy of retinal pigment epithelium and choroid. Depending on which photoreceptors, rods and/or cones are active, environmental or background illumination can be characterized as scotopic, which is equivalent to the illumination provided by starlight, in which only rods mediate vision; mesopic, which is equivalent to the illumination provided by moonlight, in which both rods and cones mediate vision; and photopic, which is equivalent to the illumination provided by indoor lighting and sunlight, in which only cones mediate vision. We'll begin with the functional differences between the rods and the cones. Rods are highly sensitive to light, and a rod cell can respond to a single photon. Cones are less sensitive to light. Rods have highest sensitivity in scotopic conditions, in which cones do not respond. And cones have the highest sensitivity in photopic conditions, in which rod visual pigments are saturated and cannot respond to a light stimulus brighter than the background. And cones can adapt to a wider range of background illumination as compared to rods. Cones have higher spatial and temporal resolution as compared to rods, and visual acuity of the rod system is 10 times worse than that of the cone system. Temporal resolution refers to ability to detect a flickering light source as flickering rather than steady. Cones mediate color vision, but rod vision is achromatic. Phototransduction is the conversion of light energy to electrical impulse, which can be transmitted to the brain. Before discussing the phototransduction process, we need to understand the dark current, which occurs continuously in the photoreceptors in a dark environment. So dark current is a continuous current in the outer segment and inner segment of the photoreceptors in the absence of light stimulus. In the dark current, sodium and calcium ions enter photoreceptors through cationic CGMP gated or CNG channels in the plasma membrane of the outer segment of the photoreceptors. The sodium ions are then extruded from the inner segment by sodium potassium ATPase, and the calcium ions are extruded from the outer segment by calcium, potassium and sodium exchanger, which also requires metabolic energy. This dark current thus results in a constant state of depolarization of the photoreceptor with steady release of glutamate from the synaptic terminals of the photoreceptors in the dark environment. So counterintuitively, the photoreceptor cell is electrochemically active in a dark environment in the absence of a light stimulus. The sequence of phototransduction has been studied in detail in rods, and we are going to discuss it. However, the phototransduction process in the cones is said to be similar. The sequence starts by photon being absorbed by rhodopsin, which, as we have discussed in the previous session, is a conjugate of rhodopsin and 11-cis retinal. This photon absorption changes 11-cis retinal to all-trans retinal, which produces a conformational change in the rhodopsin to metarhodopsin 2. All-trans retinal then detaches from the opsin and is regenerated into 11-cis retinal in the retinal pigment epithelium, which we will be discussing in the next session. As we have mentioned, rhodopsin is highly sensitive to photon, and a single molecule of rhodopsin can initiate the phototransduction sequence after stimulation by a single photon. Metarhodopsin 2 then interacts with the coupled heterotrimeric G protein called transducin. Transducin exchanges a bound GTP for a GTP and dissociates into a T-alpha GTP fragment and a T-beta gamma fragment. The T-alpha GTP then activates a CGMP phosphodiesterase by removing its inhibitory gamma subunit. This cyclic GMP phosphodiesterase hydrolyzes the cyclic GMP, which gates the CNG channel into 5-guanosine monophosphate. As a result, there is a decrease in cyclic GMP levels, which close the CNG channel, as a result of which the dark current decreases and the cell hyperpolarizes. And this hyperpolarization leads to decrease in glutamate secretion from the synaptic terminals of the photoreceptor. So again, counterintuitively, in the presence of a light stimulus, The CNG channel closes and the cell becomes electrochemically less active. Amplification of response to a photon can occur by a single metarrhodopsin molecule being able to activate another transducin molecule after an activated transducin molecule dissociates from the metarrhodopsin, and also by one activated cyclic GMP phosphodiesterase molecule being able to hydrolyze many cyclic GMP molecules. Signal deactivation is required for the photoreceptor to return to its resting state and maintain its high sensitivity to light, and it occurs by inactivation of metarrhodopsin, inactivation of cyclic GMP phosphodiesterase, and cyclic GMP resynthesis. Inactivation of the metarrhodopsin molecule occurs through phosphorylation of the rhodopsin molecule by rhodopsin kinase enzyme, followed by binding of arrestin to phosphorylated rhodopsin to fully quench rhodopsin so that it again becomes sensitive to photon. Rhodopsin kinase is stimulated to phosphorylate rhodopsin by removal of its inhibition by recovery, which we will be discussing further. Inactivation of cyclic GMP phosphodiesterase occurs through intrinsic GTPase activity of the T-alpha subunit of transducin, which hydrolyzes its associated GTP to GDP, thus removing the activation signal of phosphodiesterase. Cyclic GMP resynthesis from 5-GMP is mediated by guanylate cyclase, and fall in calcium concentration due to closure of the CNG channel activates guanylate cyclase. So resynthesis of cyclic GMP and inactivation of cyclic GMP phosphodiesterase increases the intracellular concentration of cyclic GMP, which in turn opens the CNG channel, and inactivation of metarrhodopsin by rhodopsin kinase and arrestin restores the sensitivity of rhodopsin to photons. Light adaptation is the process by which sensitivity of photoreceptors to light stimuli is maintained with increased background illumination of environment, and it is measured by assessing the incremental threshold. Incremental threshold is the minimum intensity of a light stimulus over and above the background illumination which an eye can detect. With increase in background illumination, this incremental threshold increases for both rods and cones. In scotopic conditions, rods show an increase of this incremental threshold, but only up to a maximum background illumination, beyond which rods become saturated and does not respond to any light stimulus brighter than the background. In photopic conditions, cones show a continuous increase of this incremental threshold with increase in background illumination without reaching a maximum background illumination, so maintains this sensitivity to incremental light stimulus over background even at background illuminations which bleach all visual pigments. And it has been seen that rods can perceive a change in light intensity of 1% of background illumination in scotopic conditions, whereas cones require a change in light intensity of 20% of background illumination in photopic conditions. Light adaptation occurs during transition from dark to bright environment, and light adaptation occurs much more rapidly than dark adaptation. Light adaptation occurs by desensitizing the photoreceptors to steady background illumination so that they can respond to a brighter stimulus, and this is mediated through the decrease of intracellular calcium ion concentration with exposure to light when the CNG channels close. Decrease of intracellular concentration of calcium leads to resynthesis of cyclic GMP by stimulation of guanylate cyclase enzyme, regeneration of rhodopsin, and also increased affinity of the CNG channels to cyclic GMP. Decrease of calcium ion concentration leads to inhibition of calcium ion dependent recovery, which leads to reduction of recovery mediated inhibition of rhodopsin kinase, so rhodopsin kinase is stimulated to phosphorylate metarrhodopsin 2. Calcium ion independent mechanisms also exist, and this is said to occur by light driven redistribution of molecules among various compartments within the photoreceptor. Dark adaptation is the recovery of sensitivity to progressively fainter light stimuli when the subject remains in a dark environment following a period of exposure to bright environment. In the retinal periphery, a characteristic biphasic dark adaptation curve is obtained as seen here in this photograph. When the subject, following exposure to bright light for 10 minutes, is exposed to a dark environment and threshold luminance is measured sequentially over time with the subject remaining in the dark environment. Initially, the threshold luminance decreases rapidly for 5 minutes, which is called the cone recovery phase, followed by a plateau phase of another 5 minutes, which is called the cone plateau because the threshold luminance here denotes the absolute cone threshold, which means that cones do not respond to fainter light stimuli as compared to this threshold. Another decrease starts which continues for 40 to 50 minutes, which is due to recovery of the rods. And the break between the two portions of the curve is called the cone rod break. Finally, a plateau is reached, denoting similarly the rod absolute threshold, with the rods not being able to respond to light stimulus fainter than this threshold. In the fovea, the dark adaptation curve is monophasic, reaching only up to the cone plateau. Because rods are absent in the fovea, so this portion of the curve is absent in the fovea, and the fovea is not able to respond to light stimuli fainter than the cone absolute threshold. If instead of polychromatic light, short wavelength light is used to assess dark adaptation, a biphasic dark adaptation curve is obtained, and when dark adaptation is assessed with long wavelength light, a monophasic curve is obtained, and this is said to be due to rods and cones having similar sensitivities to long wavelength light without any separation of their threshold luminance. But on the other hand, rods are more sensitive to short wavelength light as compared to cones, because of which the sensitivities of the cones and the rods differ with short wavelength light. Mechanisms of dark adaptation include rapid turnover of opsin molecules, which we have described, and also because of redistribution of various components of the phototransduction cascade within the separate compartments of the photoreceptors. Color vision is mediated by cones, and so mesopic and photopic vision are chromatic, but scotopic vision is achromatic. There are three types of cones, each with its specific type of opsin. S or blue cone, whose opsin has peak sensitivity to light of wavelength 430 nanometers. M or green cone, whose opsin has peak sensitivity to light of wavelength 530 nanometers. And L or red cone, whose opsin has peak sensitivity to light of wavelength 561 nanometers. Working together, these three types of cones are said to have a peak sensitivity to light of wavelength 555 nanometers, whereas rods have highest sensitivity to light of 507 nanometers. But we have to keep in mind it doesn't perceive the color of 507 nanometer because rod vision is achromatic. This will be scotopic, and this will be photopic. There are two theories of color vision. In the trichromacy theory, any color can be matched with a specific combination of different intensities of the three primary colors red, green and blue. In the color opponency theory, it is said that there are two color opponent systems made of complementary colors. Complementary colors are those colors which when mixed produce a grayscale color sensation ranging from black to white. The two color opponent systems are red-green opponency system, with red and green being complementary to each other, and blue-yellow opponency system, with blue and yellow being complementary to each other. What color opponency actually means is a particular color will never appear as reddish green, greenish red, bluish yellow or yellowish blue, but may appear as reddish blue, reddish yellow, greenish blue and greenish yellow. And as we have mentioned in the previous session, color vision is a function of the parvocellular layer providing for red-green opponency system and coniocellular layer providing for blue-yellow opponency system of the retinogeniculate pathways, which we will discuss further in this session. The genes for the opsins of photoreceptors have been located to chromosome 3 for the rod opsin, chromosome 7 for the blue opsin, and X chromosome for the red and green opsins. Congenital color vision deficiencies can be classified as dichromats who do not perceive one primary color, anomalous trichromats who have abnormality in perceiving one primary color, and rod monochromats who have absence of all cones, and which is thus also called achromatopsia. Dichromats can be protanopes who have absent red cones, deuteranopes who have absent green cones, and tritanopes who have absent blue cones. Anomalous trichromats can be protanomalous in whom spectral sensitivity peak of red cone opsin is shifted to a wavelength shorter than 561 nanometers. Deuteranomalous in whom spectral sensitivity peak of green cone opsin is shifted to a wavelength longer than 530 nanometers, and tritanomalous who have an altered spectral sensitivity peak of the blue cone opsin. We can remember this with a mnemonic PDT-RGB. RGB standing for red, green and blue, and PDT standing for protanopia, deuteranopia and tritanopia. Retinogeniculate pathways are constituted by the axons of the ganglion cells in the optic nerve, optic chiasm and optic tract, and the retinogeniculate pathways are classified according to the layer of lateral geniculate nucleus to which these axons project. Magnocellular pathway originates from parasol ganglion cells and mediate motion detection, contrast sensitivity and depth perception. Parvocellular pathway originate from midjet ganglion cells and mediate visual acuity, red-green color vision and shape determination. Coniocellular pathway originate from bistratified ganglion cells and mediate blue color vision. ATP is produced in the inner segment through Krebs cycle and in the outer segment by glycolysis, hexose monophosphate shunt and creatine phosphate pathway. NADPH produced by the HMP shunt pathway reduces all-trans retinal to all-trans retinol, which is taken up by the retinal pigment epithelium for degenerating 11-cis retinal. NADPH also reduces glutathione to regenerate glutathione in the glutathione peroxidase antioxidant system, which we'll discuss next. Retina is prone to oxidative damage because of abundant oxygen supply in the retina and the choroid. The inner segments of the photoreceptors, being mitochondria rich, produce reactive oxygen species which may leak out from the mitochondria. The membranous discs in the outer segments of the photoreceptors contain polyunsaturated fatty acids which undergo peroxidation to peroxides on exposure to reactive oxygen species, damaging these membranes and blocking protein function. The most abundant PUFA in photoreceptor membranous discs is docosahexaenoic acid, the most unsaturated among the PUFAs. Phagocytosis of disc membranes in the retinal pigment epithelium also generates these reactive oxygen species, and UV light to which the retina can be exposed can produce singlet oxygen from molecular oxygen. We may note here that cornea is able to block light of wavelengths less than 350 nanometers but cannot block UV light between 350 nanometers and 400 nanometers. But upon development of nucleosclerotic cataracts, wavelengths less than 430 nanometers, which includes all UV light, are blocked from reaching the retina. Three disease entities have been particularly linked with oxidative damage to the retina, and these are age-related macular degeneration, diabetic retinopathy, and inherited retinal disorders. Antioxidant mechanisms in the retina include glutathione peroxidase, glutathione and selenium system, which detoxifies lipid peroxides, vitamin E, which blocks the chain reaction of lipid peroxidation by scavenging free radicals, vitamin C, which blocks UV light and acts synergistically with vitamin E to scavenge free radicals, carotenoids or xanthophylls, which, as we have discussed in the previous session, are found in Henle's layer in the macula but are absent in retinal pigment epithelium. Zeaxanthin concentration is more in cone dense region in the center of the retina, whereas lutein concentration is more in the rod dense region in the periphery of the retina. Beta carotene, the precursor of vitamin A, is found only in minor quantities in the retina. Carotenoids quench singlet oxygen, blocks blue light, and reduce chromatic aberration at the fovea. Superoxide dismutase and catalase systems convert superoxide to hydrogen peroxide and then to water, and zinc is a part of superoxide dismutase enzyme and is also important for regeneration of glutathione in the glutathione peroxidase system. To recap the salient points, environmental illumination can be classified as scotopic when only rods are active, mesopic when both rods and cones are active, and photopic when only cones are active. Rods are most sensitive in scotopic conditions and cones are most sensitive in photopic conditions. Cones provide high spatial and temporal acuity and are responsible for color vision. Rod vision is achromatic. Phototransduction is the conversion of light signal to an electrical signal, and there is a continuous dark current which keeps the photoreceptors depolarized and makes them secrete glutamate from their synaptic terminals continuously in the absence of light. Upon exposure to light, photon is absorbed by rhodopsin, which converts 11-cis retinal to all transretinal, which in turn produces a conformational change in rhodopsin. Rhodopsin then activates cGMP phosphodiesterase through transducing a heterotrimeric G protein, which then hydrolyzes cyclic GMP to 5-GMP. This closes a cGMP gated cation channel through which this dark current passes, thus hyperpolarizing the photoreceptors and decreasing the secretion of glutamate from their synaptic terminals. Amplification of light signal occurs by one activated rhodopsin molecule being able to activate multiple transducin molecules and one activated cGMP phosphodiesterase molecule being able to hydrolyze many cGMP molecules. Restoration of the photoreceptor sensitivity to light occurs by regeneration of rhodopsin, inactivation of cGMP phosphodiesterase, and resynthesis of cyclic GMP. Light adaptation is the maintenance of sensitivity to a light stimulus brighter than the background with increasing background illumination. Rods and cones respond to light stimuli with progressively larger incremental thresholds with increase in background illumination in scotopic and photopic conditions respectively for rods and cones. But rods can respond only up to a maximum background illumination, beyond which they do not respond to any incremental threshold, however large. Cones continue to respond to light stimuli with larger incremental thresholds with progressive increase in background illumination. Dark adaptation is the sensitivity to progressively fainter light stimulus when a subject, after being exposed to bright light for 10 minutes, is shifted to and remains in a dark environment. When the threshold intensity of a light stimulus is measured sequentially over time in this dark environment, a biphasic curve is obtained. The first portion of this curve lasts for 10 minutes when a plateau threshold intensity is reached called cone absolute threshold, with the cones not being able to respond to a fainter light stimulus than this cone absolute threshold. After the cone plateau is reached, the second phase of the curve starts, which depicts the regeneration of rods till a plateau of threshold intensity is reached in 40 to 50 minutes, called the rod absolute threshold, with the rods not being able to respond to a fainter light stimulus than this rod absolute threshold. Color vision is mediated by cones, and there are three types of cones with their specific types of opsin proteins: the blue or S cones, the green or N cones, and the red or L cones, with their peak sensitivity at 430 nanometers, 530 nanometers, and 561 nanometers respectively. Acting together, the three cone systems have maximum sensitivity at 555 nanometers, whereas rods have maximum sensitivity at 507 nanometers. There are two theories of color vision. The trichromacy theory states that any color can be obtained by appropriately mixing light of three primary colors: red, blue, and green. The color opponency theory states that There are two color opponent systems, red green and blue yellow, and color of a light source cannot be perceived as a mix of the two colors of the same system. Three retinogeniculate pathways are classified according to the layer of lateral geniculate nucleus they project to. The magnocellular pathway originating from parasol ganglion cells mediate motion detection, contrast sensitivity, and depth perception. The parvocellular pathway originating from midjet ganglion cells mediate high spatial visual acuity, red-green color opponency system, and shape detection. The coniocellular pathway originating from bistratified ganglion cells mediate blue-yellow color opponency system. Retina is prone to oxidative damage because of exposure to abundant light and oxygen, abundant mitochondria generating reactive oxygen species, and presence of large quantities of polyunsaturated fatty acids in the membranous discs, with these polyunsaturated fatty acids being prone to damage by reactive oxygen species. Antioxidant mechanisms in the retina include glutathione peroxidase, which require selenium and zinc, carotenoids such as lutein and zeaxanthin, vitamins E and C, and superoxide dismutase catalase system, which also requires zinc. Thank you for listening.